This is an OCR A-level biology video on carbohydrates. In a previous video I looked at fats and the phospholipid bilayer, but today we're going to do carbohydrates. So let's get straight into it. So carbohydrates are insoluble in water, so they don't affect the water potential of a cell. They have a monosaccharide form and a polysaccharide form. So the monosaccharide form is your monomers and the polysaccharide is polymers, and they join together through glycosidic bonds, which is a condensation reaction. So water is produced. So two monosaccharides join together by glycosidic bonds to form a polysaccharide and water. Some monosaccharides I've got here for you: alpha glucose, fructose, beta glucose and beta galactose. And some polysaccharides are maltose, sucrose, lactose and cellulose. Maltose is formed by two alpha glucose molecules joining together. So alpha glucose plus alpha glucose makes maltose. Sucrose is alpha glucose plus fructose. Beta glucose plus alpha glucose makes lactose, and cellulose is made by beta glucose plus another beta glucose molecule. So you just have to learn that. There's no other way of telling you it. I'm just reading the information off to you. You just have to take your time and learn those. Two of those are reducing sugars, and they're maltose and lactose, and non-reducing sugars are sucrose and cellulose. And the difference between reducing and non-reducing is useful when you're looking at the chemical tests, the molecular tests for simple reducing sugars. Okay, so when we're looking at carbohydrates, we're dealing with glycosidic bonds, and to get the energy from the carbohydrates, because you should know carbohydrates are an energy source, you need to be breaking the glycosidic bonds to release the monomers. So how do we do that? It's a hydrolysis reaction. So anything formed by a condensation reaction can be broken by a hydrolysis reaction. So now I'm going to show you alpha and beta glucose. So first of all, here we've got alpha glucose. So two alpha glucose make maltose, which is the polysaccharide, or disaccharide is another word for polysaccharide. An alpha glucose and a fructose make sucrose. So this is what alpha glucose looks like. So it's a hexose sugar, so 6 carbons. We've got carbon 1, 2, 3, 4, 5, 6, and 1 oxygen here, okay, within our ring. Basically, all you need to do is learn this diagram for alpha glucose. Once you've learned alpha glucose, you will realise that this group here, this carbon, hydrogen and hydroxide group, and this group here are symmetrical. Okay, so the molecule is pretty symmetrical. I know that these two aren't the same, but just learn this molecule and remember what these look like in alpha glucose. Let's compare that to beta glucose. So first of all, I'm going to draw out alpha glucose again, and you might question why, but this helps you realise how subtle a difference there is between the two molecules. So this is an alpha glucose molecule, exactly the same as this end. So now the only thing we change is this. We erase that and switch them upside down. So let's highlight that. Let's highlight our differences. Okay, so here on the left in our alpha glucose, we've got our hydrogen on the top and our hydroxide at the bottom, symmetrical to the left group. In the beta glucose, everything else is the same, but the only thing we change is these two flip upside down in our beta glucose. So our hydroxide group ends up on the top and our hydrogen ends up at the bottom. I'm not going to spend any longer on that. It's just a matter of learning the diagrams and just keep practicing them and going through them. So our beta glucose is opposite but our alpha glucose is symmetrical. It's one sort of way you can remember it. Not the whole molecule, it's not symmetrical. Don't write that in an exam, but just how to remember these two groups. So glucose is C6H12O6. Also remember that. So if you're ever asked to draw these, just to check that you've got the right number of atoms, just remember C6H12O6, and that should be how many of each atom you have. So now I've got ribose and deoxyribose. Ribose is both of these are pentose sugars, so it's a 5-carbon sugar. They have a different formula, so they have C5H10O4. Once again, there is only one subtle difference between the two. So ribose is here. I draw it out again. Then we remove this hydroxide group and replace it with a hydrogen. And that is the only difference between the two molecules. Once again, just learn the two molecules. They'll be in your textbooks as well. Okay, so now we've looked at some of our monomers, our alpha and beta glucose. We can now look at some carbohydrates. So our cellulose, glycogen, amylose and amylopectin. Amylose and amylopectin form starch, and I'll write that on later. But first of all, let's look at cellulose. So cellulose is found in plants forming cell walls. It is tough, insoluble and a fibrous substance. It's formed of beta-glucose molecules which bind together to form straight chains. Each beta-glucose molecule is rotated 180 degrees alternatively, and they have beta 1 to 4 glycosidic bonds. So the carbon 1 and carbon 4 bind together. So this is carbon 1, that is carbon 4. If these two molecules bind together, if they're both alpha glucose, that will be where your glycosidic bond is formed. So each is rotated by 180 degrees. The hydroxyl groups on carbon 2 stick out, enabling hydrogen bonds to be formed between the chains. So cellulose is an excellent material for plant cell walls. When we get 60 to 70 cellulose chains bond together, they form microfibrils, about 10 to 30 nanometers in diameter. These then bundle together to form macrofibrils, containing up to 400 microfibrils embedded into pectins to form cell walls. They run across all directions of the cell wall for extra strength, and the hydrogen bonds between our cellulose chains adds even more strength. There's space in between the microfibrils to allow water and mineral ions to pass on their way in and out of the cell, making cell walls fully permeable. At the end of the video I'll show some exceptions to that for cell walls, but let's just get on to glycogen now. So glycogen is stored in the liver of humans, and it is made up of alpha glucose molecules. So whereas we had beta glucose in cellulose, we've got alpha glucose in glycogen. So alpha 1 to 4 glycosidic bonds, which we see 1 to 4 is going to come up in all of these. So carbon 1 and carbon 4 always bind together to form chains, okay? And these bonds are short, and the importance of that being short will be clear when we compare glycogen to amylopectin. So these short bonds give glycogen a tendency to coil. We've also got alpha carbon 1 to 6 bonds, glycosidic bonds, forming side branches, and these side branches make it easier to remove monomer units as there are more ends. Chains are formed by 1 to 4 glycosidic bonds, but branches are formed by condensation reactions between carbons 1 and 6. Okay. Glycogen's branched alpha glucose chains are compact, so they don't occupy large areas. The glucose molecules can easily be snipped off the chains by hydrolysis, which is catalyzed by enzymes. Amylase hydrolyzes 1 to 4 glycosidic bonds, and glucosidase hydrolyzes 1 to 6 glycosidic bonds. The ability to be stored in large amounts and the ability to so easily lose glucose molecules makes glycogen the best energy source in humans. Okay, the core of our glycogen is called the limit dextrin, and that is indigestible, but it has a use in mucus, so it's the main sort of component of mucus. So now amylose, once again, alpha glucose, and the only one that is beta glucose is cellulose, and that is the only one that's not really a storage source of carbohydrates. It's just for cell walls, okay? So all the storage ones involve alpha glucose with alpha 1 to 4 glycosidic bonds, but the bonds, I wrote long here, just ignore that. At the end of this I cross it out. You don't need to know how long the bonds are in amylose. They coil into a spiral or helix. The 3 hydroxyl groups found on the inside of the double helix, allowing the molecule to be less soluble and more hydrogen bonds form. So amylose chains are unbranched. They do not have that 1 to 6 glycosidic bond, and this double helix is important when we look at testing for molecules. Iodine gets trapped in the middle of the helix, allowing starch to be tested for. Okay. So amylopectin is found within cells within large starch granules, commonly in the palisade layer, just like amylose. Like glycogen, has both carbon 1 to 4 and carbon 1 to 6 glycosidic bonds between alpha glucose molecules. Like amylose, it coils into a spiral shape, held together by hydrogen bonds, but with branches instead emerging from the spiral. As the bonds are the same as those within glycogen, you'd expect it to behave in the same way. However, the bonds in amylopectin are longer than those in glycogen, giving amylopectin a tendency to coil. It's another major energy store, while it is branched like glycogen, so it could be accessed very easily. But like glycogen, it's got this limit dextrin, so not the whole thing can be digested. So the two ones in red here, our glycogen and amylopectin, are good stores, as they've got these 1 to 6 glycosidic branches, which can easily be accessed and the monomers can be cut off. But they've both got this limit dextrin, and the only real difference between the two is the length of these 1 to 4 glycosidic bonds. Glycogen has short bonds, so it's got less tendency to coil, whereas amylopectin has long bonds and it's got a greater tendency to coil. So glycogen is slightly more compact than amylopectin, but they're both good forms of getting energy quickly. Finally, as I've started writing here, hydrolysis of glycosidic bonds breaks disaccharides. Amylase hydrolyzes carbon 1 to 4. Glucosidase hydrolyzes carbon 1 to 6, which I've already mentioned to you, but it's just written there for you now. And one more thing about cellulose. So another few characteristics of cell walls. Cellulose gives it a high tensile strength, so it prevents the cell wall from bursting, and that's a really good property in plants, as when they're getting turgid and the cell membrane is pushing against the cell wall, it does not explode. Okay, so lignin, which you remember from xylem vessels, cutin and suberin, block spaces in cell walls to make it waterproof and make it impermeable. Okay, so cellulose cell walls can be permeable if they do not have these waxy substances inserted into them, but they can also be adapted to be impermeable, okay, and waterproof. One final point I'm going to make here is, as we saw with lipids, polysaccharides or disaccharides are also less soluble in water, meaning they do not affect the water potential of the cell. So that's another adaptation that makes them good for storage. By being non-soluble, you do not affect the water potential of the cell. So that was all on carbohydrates. Next video we are going to be looking at proteins, and after that we'll look at inorganic ions within the same video. and in a separate video we're going to look at the biological tests for biological molecules, including colorimetry, biosensors and chromatography.